Okay, how's everybody doing? We just have um, like it seems like we're just seeing the light at the end of the tunnel here, and the onslaught of the believers in evolution trying to impose their doctrine on us. So let me just show a little thing here. I have a little thing to show. It's of a lecture from somebody from Yale, and okay, so his name is, let me see, his name is, oh, Haifan Lin. And if you would like, you can watch the entire thing. It's the Pee-wee piRNA pathway, a new paradigm of gene regulation with Haifan Lin. It has 4,000 views and only 7 comments. And kind of irrelevant comments. Some of them say, oh, great lecture, whatever. But anyways, this is like, maybe it hasn't sunk into the scientific community yet, but let's go ahead and learn something before they can. And this thing is from last year, basically, May 17, 2019. So I'll just give you a little picture of it. You can watch the rest of it on your own. It's really good. It's an hour long. Of course, I'm not going to do that. But check this out.
So far, I've told you about when PV molecules and piRNAs are in the nucleus. That's one of the major functions. Then how about when these proteins are in the cytoplasm? Amazingly, they have another function that's completely unexpected as well. So as we know, our genome contains 23,000 genes. In addition, we have vast intergenic regions in which about 1 million copies of transposons reside. And 96% of these transposons in the human genome are retrotransposons. They transpose by making these intermediate RNAs. Transposons are today still generally viewed as the selfish parasites that invaded our genome. We cannot get rid of them, just have to cope with them. There are a few studies talked about transposons function in mutagenesis and potentially transcriptional regulation, but the genome-wide function of transposons largely remain unknown. Now, because of many labs work, including that of mine, we know this intergenic region also contains new types of genes which encodes non-coding RNAs. So these non-coding RNA coding genes will code for many link RNAs and millions of piRNAs. And finally, we should not forget that in our genome, we also have the abundant existence of pseudogenes. In the human genome, we have 14,000 pseudogenes. Most of the pseudogenes are still expressed and made into messenger RNAs, or sometimes from the antisense promoter into antisense RNAs. However, nothing is known about the function of these pseudogenes. They're in general regarded as the defunct carcass of their active genes on their way out during evolution. Of course, very little is known about the functional or regulatory relationship among these major constituents in the genome. Today, I hope to illustrate to you that transposons actually play a very important role in regulating the expression of protein coding genes at the post-transcriptional level. In addition, I'll show you that many pseudogenes also play a very important role in regulating the expression of their sister sibling genes. Furthermore, I'll illustrate to you that transposons also play a very important role in regulating the expression of many link RNAs. And finally, as you might have guessed, I hope to demonstrate to you that all these regulations are mediated by the PV pathway.
Okay, so we got a lot more going on there than what we have been led to believe. Obviously, they haven't been telling us the whole truth and nothing but the truth. Obviously, they've assumed that a lot of the genome is junk from so much reproduction and everything. And of course, because they assumed their own um, narrative of things evolving. And so if things evolve, then they're going to have a lot of junk that's not even useful because of all the mutations going on. Turns out that most of it, probably nearly all of it, is essential and useful in many different ways. And he's going to get into how some of the imbalances cause cancer. So we're just scratching the surface now of all the function that's in the DNA because the evolutionary paradigm has put us way behind on knowing what the facts are. So if you see an evolutionist, you can goad him a little bit that everything they ever predicted was all wrong.